Namaste, one and all. I welcome everyone on this second day of our transitional Ayurveda lecture series. Today, we have a very distinguished speaker, Dr. Sarika. Dr. Sarika is a scientist and a product manager with a PhD degree in biotechnology and having over 12 years of experience in biotechnology, clinical research, and integrative disease biology. Her expertise lies in AI driven drug discovery, including target validation, drug repurposing, and clinical trial prediction, with a focus on oncology, rare and metabolic diseases. Dr. Sarika is skilled in literature mining, biological data curation, and cross functional collaboration, and has a strong background in managing research projects, authoring clinical trial and documents, and developing scientific strategies. With this very brief introduction, I wholeheartedly welcome Dr. Sarika in this lecture series. So, uh, without any further delay, we will start our session. So, the mic is to you, Dr. Sarika. You may please share your PPT and you can start.
Am I audible?
Yes,
Hello?
madam. Yes, madam.
Okay, okay. Sorry, I got on unmute. So,
Yeah. Please share your PPT, madam.
yes, sure, sir. Sir, I would like to thanks All India Institute of Ayurveda for organizing this such a nice event and for the celebration of the Ayurveda Day. And I want to special thanks to all the organizers of the All India Institute of Ayurveda, Dr. Mohan Joshi sir, and I also want to thanks Dr. Supriya Bhalerao ma'am because she gave me this opportunity to express myself and the knowledge of gut microbiome in front of you. Thank you, sir. I'll just share my screen.
Yes, madam. The screen is visible, madam, and your voice is also very clear.
Yes, sir. I'll just. Is it okay? Is it visible?
Yes, ma'am, it's visible, ma'am. Just if you could do the full screen, that will be
Yeah, yeah. I'll do that. Okay. So
exactly.
today, yeah. Am I audible, right?
Yes, ma'am.
Yes. So today we will be discussing on the topic that is a gut microbiome. As we all are aware about in Ayurveda, we have like there are concept called as Agni, that is a digestive fire, right? So and as it was mentioned in Ayurveda that this Agni is important for the maintenance of the health and for the cause of the disease. So here we are talking about the gut microbiome, how this gut microbiome is important for the health as well as in the diseases. So in the today's discussion, we will be covering on the overall gut microbiome, its diversity, its function, and its impacts on the overall gut microbiome. Okay, so we will start our discussion with the very basic details about the microorganism. So as we know, these microorganisms are nothing but the small microscopic tiny organisms that cannot be seen with the naked eyes. And this microorganism have a various or the large diverse range of the form. Like for example, some are circular, some are spiral, some are y-shaped. So the morphologically they are very different, and these microorganisms are also present in the different kind of environments. Like for example, if you go to soil, water, air, everywhere you will get these kind of microorganisms. So when we talked about the microorganism, these are nothing but the small living things which are present throughout the system or throughout the surroundings. When we talked about the microbiota, this microbiota refers to the community of the microorganism that lives in the specific habitat. Like for example, today in the today's discussion, we will discussing on the gut microbiome. So generally we talk this as a gut microbiota. Then this microbiome is nothing but the collective genetic material of the microorganism, which mainly includes bacteria, viruses, fungi, and all other type of microorganisms or the living system that can present inside the body or that can live, like for example, skin surface. So this is kind of a microbiome. So the overall complex community of the microbiome or the microorganism is called as a microbiome. So why we are talking about the microbiome? Because as I have mentioned, there are certain microorganisms which can be detected with the advanced techniques, like for example PCR, 16S sequencing. But there are certain uncultured microorganisms that also play a very important role when we talked about the gut microbiome. So that's why overall complex community of the microorganism is called as a microbiome. So let's talk about the human microbiome. So, when we talked about the human microbiomes, so there are different anatomical sites on which these microbiomes are present. For example, the skin. Skin is the body's largest organ where we can find a vast variety of microorganisms, and these microorganisms not necessarily every time are responsible for the cause of infection, but in certain cases these microorganisms or the skin microorganisms, they are helpful or they act as a protective microorganism. Therefore, in the any anatomical site, the balance of the microbiome is very important. Next we talked about the oral microbiome. So this mouth is another rich organ where the microbial diversity is high as compared to the other anatomical sites. And this oral microbiome is also very important in maintaining the health and for the cause of the various diseases. For example, there are dental caries or the dental conditions in which this oral microorganism plays a very important role, because these organisms are responsible for the digestion of the food, for the dental caries, then for metabolism of the certain compounds. So therefore this oral microbiome is also play a very important role. Next is the sexual organ microbiome. Here also microbiome of a various sexual organ is essential for maintaining the healthy uh, person or the healthy balance of the different this thing. So this microorganism also helps in the protection of the harmful pathogens. These microorganisms are also helps in the maintaining the pH level. They support overall growth and the overall health of the reproductive system. So therefore the dysbiosis of this microbiome from any anatomical site can lead to the diseases. So next we will talk about the gut microbiome. As we know, the gut is a major hub of microbiome or the microbial diversity. So we will discuss the gut microbiome in detail, but these gut microbiome play a very significant role in various biological processes. The microbiome imbalance or the dysbiosis in the gut microbiome or any anatomical site microbiome can lead to the cause of the various disease conditions. So as we all are aware, there are certain reports and the recent publications they are suggesting the dysbiosis in the microbiome can lead to the obesity, GI related disorders, some mental related disorders. So it is well proven that dysbiosis in the gut can lead to the disease condition. So, by understanding these different areas of the human body and their associated microbiome, we need to understand the intrinsic relationship between these microbiome and the overall health. So, in the today's discussion, we will be significantly focused on how this gut microbiome plays important role. Okay, so with this, we will just discuss this gut microbiome. As we know, this gut microbiome composition is very complex, which consists of trillions of bacteria, which includes bacteria, fungi, protists, all other kind of living system. This gut is not a uniform environment. When we talk from the stomach to colon or the rectal part, so there are different environmental condition present. So at different environmental condition, we have a different what we can say different species and the variety of microorganism present throughout the digestive system. Like for example, when we talked about the stomach, the acidic pH is there. At the acidic pH, only the bacteria which can sustain at the acidic condition, like for example lactobacillus, streptococcus, which will get sustained. As we go down to the duodenum, jejunum, the pH level will go on increasing and the colonization of the different type of bacteria will come there. In the colon, the pH is like somehow neutral, that is 7. So at the colon level, we have a bacterotides, eubacterium, these kind of organism. So when we talked about the overall microbiome, it is very complex ecosystem of the microorganism. And when we consider a healthy microbiome or the healthy adult individual, which consists of approximately 100 trillions of bacteria in gut alone. So, for example, when we consider or when we compare this with the human cell, this is 10 times larger or 10 times more than the actual cell type. And as we know, in the human, we have a 23,000 genes, which actually codes for the some kind of the functional protein. But when we talked about the microbiome contribution, it is approximately 33 lakhs. So you can imagine how this microorganism contribute to the overall health and disease. As when we, as a doctor, when we say about there are 23,000 proteins which are present, or not present, but these are actively expressed and which are important in the uh, cause of the disease or maintenance of the health. But this microbiome contributes more than that. So you can imagine how vast this microbiome is. This microbiome is also recently recognized as a hidden organ, because in the earlier anatomy, it was not mentioned that a microorganism is an organ. And currently, new therapeutics, which are considered this microbiome as a therapy type, and this is also called as a hidden organ or the second brain of the human body. So that's why these microorganisms are very important and play a very important role, which perform a very vital functions, like for example digestion, immunity, certain neurological conditions like mood, sleep. These pattern also got affected or maintained with the healthy microbiome. Is it okay? Any questions so far? It will be good if we can have a interactive session. Any questions so far?
No, not posted.
Okay.
Audience, any questions? You can post questions in the chat box.
Okay. Fine, sir. Sir, it will be great if we can have an interactive session, because while explaining, if the participants are having questions, so I can answer immediately.
Sure.
Okay. So moving ahead, as I mentioned, this microbiome is important, but there are n number of factors which can influence the composition and the functions of the microbiome. So in this slide, we will be particularly discussing on the what are the different factors which are actually affecting the microbiome composition. As a doctor, as we all know that this microbiome is varying from the birth to death, right? So this microbiome evolves throughout the lifespan. Like for example, during the c-section delivery or the during normal delivery, based on the type of delivery or the mode of delivery, the microbiome composition is vary. Then based on the breastfeeding condition, if the child is having breastfeeding or not, based on that, again the microbiome composition go on varying. Another most important condition is the diet is very important condition, where the microbiome composition can get varied. And as we know, this diet play a very crucial role in shaping the health or the maintaining the health. And because this diet is also important for the maintenance of the microbiome. That is the reason why diet is an important type. So what type of food we consume, that determine the availability of the nutrients to the microorganism, and based on that the microbiome composition will get maintained in the gut. For example, I'll give you, if a diet which is rich in fiber, fruits, vegetable, which can promote the health of beneficial microorganism. If we have a junk food, then which can promote the growth of the harmful microorganism. So that is the reason where diet plays an important role. Another important factor is the exercise. So there are recent publications which can strongly say that exercise can modulate the microbiome. Like for example, if you have a healthy lifestyle, you are doing a routine exercise, your microbiome can go on the normal side, your microbiome is maintained, and this regular exercise also proven to show a reversal of the diabetes and the many metabolic conditions. So therefore healthy lifestyle is also play a very important role. Another factor is genetics. As I have mentioned, genetics is also important. Like for example, some people are having food allergy, that is depend on the genetic constituent of an individual. So therefore this genetic is also important for the maintenance and the sustenance of the microorganism. Another important factor is the aging. Like as we started with the birth mode, so as the people or the person go on aging, this microbiome composition will go towards the dysbiosis side, because during the aging it was observed that immunity just go down, and that is the reason the dysbiosis happen. And there are certain reports also which can suggest that during the aging process the microbiome get dysbiosis. Another important factor, that is a drug intake, or the some kind of antibiotic which usually take. So this antibiotic or any kind of the medication generally cause the resistance to the bacteria, and when we are treating any kind of the bacterial infection, they can have a significant impact on the gut microbiome composition. For example, these drugs whatever we are taking for any condition, these not only kill the harmful bacteria, but they can also hamper the beneficial bacteria. So therefore the prolonged use of this antibiotic or any kind of medication can ultimately lead to the dysbiosis, and which can lead to the imbalance of the microbiome. Another important condition is the geography, but this condition is also important. Like for example, if you go and do the microbiome analysis from the Indian population, the microbiome is different than the US population. So therefore geography also plays an important role for the microbiome. But these factors, like for example geography, cannot be changed, right? So this is natural, we have to accept that. But all other things, like for example geography, aging, genetics, that we can't change. But there are other factors which I discussed. For example, environmental exposure, that also we can just avoid to go into the pollution or the some other environmental exposures which can have a dysbiosis in the gut. So you can imagine there are certain modifiable and the non modifiable risk factor which can lead to the dysbiosis of the gut. I hope it is clear. Okay, so next we will move towards the impact of the gut microbiome on, yes, sir, impact of the gut microbiome on the overall health. So, as we know, this gut is important for the digestion. While digestion is a significant function, this microbiome are having a n number of other impact and the other what we can say functions in the human body that play a very important role. So first, let's talk about the immune system. As we all know, this immune system play a very important role in the maintenance of the health. So when we talked about the immune cell, so how many percent of the immune cells which are present? So approximately 70% of the immune systems are resides in our gut. So this gut microbiome play a very important role in development and maturation of our immune system, particularly in the childhood. If we can have a good healthy lifestyle, good diet, then the immune system is stronger as compared to the children who are having the uh, not healthy lifestyle, not a good proper food. So therefore this immune system is also managed, or what we can say, monitored by the gut microbiome. Then we can have a different function, like for example regulation of the intestinal motility. That is also very important, because there are certain uh, diseases which are associated with the intestinal motility, IBS. Then there is a metabolism at the weight regulation. So these gut microbiome also play an important role in the obesity. If we have a gut dysbiosis, that can lead to the obesity, diabetes, and the metabolic condition. We can discuss in detail with the gut brain axis as in the future slides. So there are another function, like for example drug metabolism. I have already mentioned about how this drug intake is having the impact on the gut microbiome. Then barrier function and the gut integrity, that is also got maintained, and with the good microbiome or the good microorganism. Then production of the vitamins and the short chain fatty acids. So these vitamins and fatty acids are also having some positive impact, like for example SCFA, that is a short chain fatty acid. These are the bacterial metabolites. This play a very important role. So other than the digestion, we have a n number of different functions which can contribute to the overall health. And if we have any dysbiosis or the any imbalance between these functions, which can ultimately lead to the disease condition. Okay, so let's discuss about the gut dysbiosis. So as we discuss how this gut microbiome are very important, so this dysbiosis is nothing but the change or the disruption of the whatever healthy microbiome is there, and this is very crucial for the health. This gut dysbiosis, that is the disturbance of the balance which can leading to the negative consequences of the health. Basically this gut biosis, when we talked about the infectious diseases, so if we have a dysbiosis in the gut, the majority time, as I have mentioned, this dysbiosis is having the what we can say role in the immune system. If we have a dysbiosis, it will hamper your immune system, and ultimately the infection possibility or the cause of infection or the ratio of the infection is high. So therefore this gut dysbiosis is what we can contributed to the infectious diseases. When we talked about the cardiovascular diseases, in the cardiovascular diseases, like for example hypertension, atherosclerosis, here also it is well proven that dysbiosis in the gut can lead to the hypertension, because it is ultimately leading to the metabolism. Like for example there are metabolic disorders also, obesity, type 2 diabetes. This is also because of the dysbiosis. Then there are certain condition, like for example autoimmune disorder, cancer. So you can see the vast spectra of the diseases. So you can consider any organ from your human body. All the diseases which are associated with these organ are ultimately at what we can say ultimately causing the or the responsible because of the gut dysbiosis. And these all research, at least cause of these indications or the cause of the diseases are because of the gut biosis. This is well proven in the recent literature. Okay, so let's discuss about the gut brain axis. As I have mentioned, this gut brain axis is very important when we talked about the gut microbiome and its role. This gut brain axis is a bidirectional communication pathway between the gut and the brain. As we know, this gut brain axis is a very exciting field of research. As we in the earlier slides we discussed how this gut microbiome is important in the mental disorder, then some metabolic disorder. So let's understand this intrinsic relationship between the gut and our brain. So when we talked about the gut and brain, this is very important that gut microbiome is having like for example the microorganisms which are present in the gut, they are not only present as a microorganism, they also responsible for secretion of the certain metabolites, like for example peptides, some microbiota derived products, some gut hormones, neurodegenerative substances. So these are the secretions. These secretions will get involved in the various biological pathways, like for example immune system, neuroendocrine system, circulatory system. So all this pathway through this secretion of the microbiome, which can lead to the different kind of the health related processes, and ultimately if we have a balance or the imbalance between these, this can lead to the some kind of the disease condition. So your health is ultimately, directly, indirectly, is linked to your gut. So your gut is, what we can say, is very basic thing which we can, as a doctor, you all are aware that in Ayurveda also we generally treat any kind of condition, any kind of disease first with the diet or first with the Agni, right sir?
Yes,
Yes,
madam.
yes,
Yes,
of
madam.
course.
Yes. So, we'll discuss in detail how this role of gut microbiome in neurodegenerative. As I thought, I'll just discuss this use case as a neurodegeneration, how this gut microbiome is important in the neurodegeneration. So, as we know, this neurodegenerative diseases mainly which consist of the Parkinson's and the Alzheimer's, and the recent research which suggests that disruption of this gut microbiome can contribute to the development of and the progression, sometimes not necessarily development, sometimes this is the, what we can say, we have this condition but which will get progressed as we have a gut dysbiosis. So first, this kind of conditions start with the various factors, as I have mentioned in the earlier side, like for example drug, then your lifestyle, your diet, your age, then your mood is also one of the important factors. Then these can lead to the gut dysbiosis. So these are the factors which can lead to the gut dysbiosis. And if we have a gut dysbiosis, which can lead to some kind of the metabolite secretion and pro-inflammatory molecule. So this pro-inflammatory, this Alzheimer is also an inflammatory condition. Like for example, it is kind of a prolonged inflammation throughout the body, which can lead to certain kind of the hormone secretions, some metabolite secretions which can cause this inflammation. And then these molecules or these secretion by the gut microbiome, which can lead to the blood brain barrier disruption. And in the earlier side we discussed that gut brain axis, that is very important for the maintenance of the overall health. So with this, throughout the process, this blood brain barrier got disturbed. So once we have this disruption, blood brain barrier disruption, it will lead to the aggravation of the oxidative stress. And once we have a oxidative stress, then it will lead to the neurodegeneration. So you can imagine how this process is complex. So not only one parameter, but it start with the various factors which can lead to the dysbiosis, and through gut dysbiosis it can lead to the neurodegeneration. So we can imagine how the gut dysbiosis is, what we can say, causative factor for the various conditions like neurodegeneration. I hope it is clear. Okay. Any questions so far?
We recommend participants to ask questions in between also, because that will bring the more interactivity.
Right, right. So I
We
was
can see
expecting
one
that.
question coming up uh,
Mm-hmm.
in the chat box.
Mm-hmm.
Two questions, I hope. Ma'am, can you just access from your side, chat box?
Yes.
On the right lower corner, you can see chat.
Right. What are the pathological investigation for gut microbiome derangement? Right.
Yes, ma'am. Are you going? If you are going to cover it in the next part, then we can continue with the presentation, and then we can come back, because I think that this part is yet to be covered.
Okay.
Anyhow, ma'am, it's up to you. You can take
Okay.
it in between, or you can just continue the flow of the presentation, and then we can come back and have a discussion.
Okay, so as we understood how this gut microbiome are important for the cause of the diseases, so we can move ahead how we can consider the gut microbiome as a therapeutic target for modulating the gut microbiome for the management of the diseases. So, for example, there are different metabolites, as I have mentioned, that dysbiosis or some harmful bacteria which can cause what we can say certain metabolites or certain hormones which can lead to oxidative stress, which can lead to the inflammation. So here we can just consider some of the beneficial metabolites as a therapeutic intervention, which can also help in the management of the diseases. Like for example, this SCFA, some steroid hormones. So these kind of certain metabolites can considered as a what we can say therapy type. Then in the this FMT, when we talked about the FMT, that is a fecal microbial transfer. What happened in the FMT? Generally, the microbiome from the healthy individual is got transferred to the disease condition or the disease individual. So this is what we can say well accepted intervention currently working, where the dysbiosis will get maintained or will get modulated with the fecal microbiome transfer. Here we have to make sure that the microbiome is in the from the healthy individual. So this is for the maintenance of the gut microbiome. So there are certain antibiotics treatment also given for the management of the or the what we can say degradation of the harmful bacteria. So there are certain probiotic treatment also given for the microbiome. So next is the microbiome based therapeutics. So as I have mentioned, this fecal microbiome transfer, this each and every therapy type, they have their own positive and the negative sides. When we talked about the fecal microbiota, so basically we have to get an fecal microbiome from the healthy individual. So transfer of the intact community in the intact community of the microbiome is little challenging. Then this is kind of screening of the patient for the healthy microbiome, because considering the current environmental condition, finding a healthy individual is a little challenging. Then diet and the prebiotic. So diet and prebiotic, like for example, we can consider some kind of the herbal intervention which can act as a prebiotic. This prebiotic are nothing but the some kind of the supplement which can modulate, which can provide as a substrate which can modulate the gut microbiome. So this diet and probiotic is very easy to go therapy type, but there are challenges for this diet and prebiotic therapy type. This diet and probiotic, they can have a very limited time impact. If we have a long term impact, the continuous intake of this diet and prebiotic should be there. Then symbiotic microbial consortia. So there are certain probiotics consortia and the supplements which are currently available in the market, which we can consider as a supplement, and this symbiotic microbial consortia can be considered as a therapy type. Then we have a engineered symbiotic bacteria. So as we are advancing with the science, so there are certain engineered symbiotic bacteria. So these engineered symbiotic bacteria are nothing but the some kind of the engineered microorganism where the positive or the positive contributing proteins are present, and these can be act as a therapy type. So there are microbiota derived proteins and the metabolites. So these are nothing but the some kind of the metabolites which are directly derived from the microorganism, and directly as a supplement we can give this protein metabolites or some hormone. So this is the recent development in the microbiome therapeutics. So this is the first FDA approved drug, that is SCR1, that is the first oral drug which is approved to treat the microbiome. And this first FDA approved drug is basically used for the recurrent infection of the Clostridium bacteria. So you can imagine how this microbiome, as we know, there are different therapeutic areas. Like for example, we have a neurodegenerative oncology. So this microbiome is also emerging as one of the therapeutic area. So based on that, this is the recent development. Then we can think of the future direction, how this microbiome is important. Like for example, we can have a microbiota based therapeutics, we can have a microbiome and cancer based immunotherapy, we can have a precision microbiome engineering. This precision microbiome engineering we detail, like for example, there are recent publications that precision medicine is also described in Ayurveda, like Prakriti analysis, that is kind of some personalized kind of analysis of an individual. So here, this precision kind of microbiome engineering is there. Then microbiome based vaccine, that is also in the development stage. Microbiome based derived therapeutics, that is also there. Then some microbiota targeted antibiotics. So these are the future direction. So first, I just want to connect this microbiome with the Ayurveda perspective. So that's why I added few slides here. Like for example, in Ayurveda, we have a Nidan, that is a diagnostic. So this microbiome analysis is also important in the diagnosis of the diseases. Like for example, in Ayurveda, we have a different concept like Ama, Agni, that will lead to the imbalance of the health or the imbalance of the system. So in modern medicine also it was related that dysbiosis or the imbalance can lead to the any kind of disease condition. So this microbiome analysis can be useful in the diagnostics. That is the first thing. Next is the therapeutic. In Ayurveda we do the chikitsa. Like for example, there are recent publications where we talked about some kind of the intervention, some kind of the procedural intervention that can lead to the management of the diseases. Like for example, few panchakarma therapeutics which are leading to the management of the diseases. And I just want to mention I got opportunity to work with Dr. Supriya Bhalerao in my earlier organization, where we have used this therapeutic intervention. Like for example, we have conducted one clinical trial where we treat the obese individual with the buttermilk. As we all know, this buttermilk is a known probiotic. So we thought we will give the buttermilk enema to the obese individual, and we have done the microbiome analysis, and we got very good result, and we published that article. So as I just want to connect this dot, how we can use this gut microbiome analysis in the therapeutics. Then we have a personalized health, that is a Prakriti analysis. So if you go to Google search, you will get a majority review article publication where we can see this Prakriti analysis is also connected with the gut microbiome connection. So in Ayurveda, generally we focus on the personalized medicine. So exploring the individual unique microbiome profile, which can help to understand the individual what we can say metabolism pattern, overall health, current condition, and which can help in the suggesting the personalized recommendation. So understanding this gut microbiome's role in overall health and diseases with the Ayurveda principle is kind of new offering or the new pathways for the holistic management of the diseases. Then I thought I'll just discuss a few part where how we can do this kind of gut microbiome analysis. So generally, this gut microbiome analysis was done with the stool sample. So collection of the stool sample is kind of some non-invasive method. We have to just collect the stool sample. We have to extract all the microorganisms present from the stool, and we do the gut microbiome analysis. Then there are certain mucosal biopsies. So during these mucosal biopsies, during the procedure, like for example colonoscopy, site-specific microbiome will get collected, and with this biopsy, again DNA will get extracted, and we can do the microbiome analysis. There are another method from which we can do the microbiome analysis, that is a lumen aspirate, where some of the fluid from the intestinal segment are extracted, or with the aspiration it will get collected and the microbiome analysis is carried out. Next is the saliva and oral swab. So this is also very important when we talk about the oral gut axis connection. But very routinely and commonly used method is the stool sample analysis. So let's explore a little about the techniques for the microbiome analysis. I know this is very technical, but we can understand what can be the ways, what can be the methods through which we can identify the microbiome composition. So as in the earlier discussion, I told you that there are only 20 to 30% of the microorganisms can be cultured. Why? Because we don't know the actual culture condition. We cannot mimic the condition which is present in the gut, which is present in the stomach, in the external environment. So there are two types of the microorganism: culturable microorganism and non-culturable organism. So those organisms which can be cultured, these are 20 to 30%. So directly with the culturing method, we can get this information. So there are some advanced method, for example, 16S RNA sequencing. The 16S RNA sequencing is one of the important method where we can identify the bacterial groups. We can identify the different genera, different species which are present in the microbiome, in the overall community from the sample, right? But the problem with the 16S RNA gene sequencing that we have very limited information. Like for example, we here with the 16S RNA, we will we will get only the genus level information, only the what we can say different genus species level information, but functional information is missing in this 16S RNA sequencing. When we go with the shotgun metagenomics, here we sequence all the DNA present. So not necessarily culturable, all culturable, non-culturable microorganisms. DNA samples will get analyzed here, and here we can get the species level information as well as the functional level information. But the shotgun metagenomics is more expensive and what we can say kind of tricky. But in generally microbiome analysis we do with the 16S RNA sequencing. So there are other advanced techniques, for example, transcriptomic analysis, metalloproteomics analysis, metabolomics analysis. In yesterday's talk, we talked about few of the metabolomics or the omics related analysis, where all the metabolites which are secreted through gut microbiome will get analyzed. In these cases, we can analyze the metabolites as well along with the active microorganisms. Okay. So I just want to highlight that while working with the Supriya ma'am in the earlier organization, we got opportunity to publish few of the articles, and like because of the ma'am only, I got a very interesting concept of the modern what we can say Ayurveda. So we published one review paper where we have mentioned that how the Ayurveda therapeutics are important for targeting microbiome. Then, as I have mentioned, we have also conducted a clinical trial where we gave the buttermilk enema to the obese individual, and we got a very interesting results there. This paper got published. And another is the symbiotic association of the gut microbiome in health and disease condition. So, for more detailed information, you can just refer this publication. Another, I thought I'll share few of the interesting articles which will help you to link the gut microbiome with the different Ayurveda concepts. So this is the publication that genomic insight into the correlation between the gut microbiome and the Deha Agni. So basically, this paper is having the link between the Agni with the gut microbiome metabolism. Another paper is like, for example, the concept from the Ayurveda, as you people are very aware about this, how this diet, sleep, and lifestyle is important in shaping the microbiome balance. Another is the how this vipaka, that is a post-digestive effect through the gut microbiome transformation. So with this, then we have a very interesting paper, as I have already mentioned, about the Prakriti analysis. So how the Prakriti analysis is linked with the gut and the oral microbiome signature. So all three Prakriti, Vata, Pitta, Kapha can have a different gut microbiome population. Then there are recent publication 2024, they can establish the link between the gut microbiome, certain dietary factors, nutraceuticals with the diabetes, right? So these are the interesting publications. Next, we talked how we can have the Ayurveda herb as a dietary factor for the microbiome modulator. Any question? Hello?
Yes,
Are
ma'am.
you-
There are questions, but not
Okay.
particularly on this.
I got
We'll
some
go through
noise.
the question answer session later, ma'am.
Okay.
You
I
can
got
just
some
continue.
noise. Yes, yes.
No,
So,
we have written down, and one by one we will go through questions, ma'am.
okay, no problem. So we can think about how the Ayurveda can, or the Ayurveda herbs can considered as a microbiome modulator. For example, Triphala. This is a very well-known formulation from Ayurveda. This Triphala is having or reported to have a prebiotic effect. As prebiotic is just kind of providing the substrate to grow the healthy microorganism. For example, Bifidobacterium and the Lactobacillus. Curcumin, this is also active metabolites which are having the inflammatory activity, and this is also a microbiome dependent activation. Then buttermilk, this is a probiotic source which can improve the digestion, which can reduce the acidity. Then ghee, which can enhance the absorption and support the SCFA production. This SCFA is nothing but the short chain fatty acids, which are considered as a positive contributor. Then there's certain millets and the high fiber diets, that is prebiotic substrate for the beneficial microorganism. So you can imagine in Ayurveda, different plants and its active constituents are uh, reported to have an various positive impact on the overall health, and how it is important in the management of the gut microbiome through uh, the various biological processes. Moving ahead, we discuss this about the therapeutic implication, like for example, personalized medicine, where we can connect the microbiome with the Prakriti analysis, and accordingly we can move ahead with the tailored therapy or the personalized therapy. Then we have a metabolic disorders. With this metabolic disorder, we can have some kind of a intervention, procedural or the therapeutic intervention for the management of these diseases. Then we have a IBD or the gut disorders, where we can use the herbs and the Basti treatment to restore the colon microbiome. Some neuroimmune disorder, where we can understand the gut brain axis modulation through diet and microbiome targeted Ayurveda. Then we have a kind of combination therapy where Ayurveda, probiotics, prebiotics, everything will work together to enhance the overall health. So this is the last part. We can think about what is the current challenges and how we can convert this limitation to the innovation for the advancement of the gut microbiome research. Like for example, so there are large number of publication we just prove or we just mentioned about the concept of the Ayurveda, linking the Ayurveda concept with the microbiome research, but very few experimental research are published. So as a Ayurveda practitioner and the modern medicine practitioner, we can have a responsibility that we have to prove this concept with the experimental evidence. So this is kind of challenges because we don't have a solid evidence to prove that this is really working. Some transient effects are there because, as I have mentioned, the long term effect with the dietary fibers or the some herbs, we have to have some constant effect. So for that, the treatment schedule should be more as compared to the what we prefer. So this transient effects, we need to think about. Some standardization related issues, like for example, as you all know, there are certain herbal formulation which vary in the quality based on the season, based on the formulation method. So we have to think about how we can resolve this issue. Then inter-individual differences, like for example, diet, genetics, geography. So we have to think how we can go ahead with this basic understanding and the basic knowledge towards the personalized therapeutics. The need of the multi-omics. This multi-omics is kind of the advanced therapeutics or the advanced technology where we can understand the hidden connections. We can do the metagenomics analysis, metabolomics analysis, transcriptomic analysis. So this is kind of challenges and the future path we can think of. Then Like for example, some to identify few biomarker which responds to the specific type of prakruti with the microbiome composition. So these, I just thought I'll just give, we will discuss this future direction where we can just think how we can uh, collaborate to prove this concept. So next, as we know, we are advancing with the science. So as AI is coming everywhere, so we thought how we can do this AI-driven Ayurveda drug discovery via the gut microbiome integration. So basic challenges when we talked about the AI based or the AI-driven discovery, that we don't have a very structured data sets with the Ayurveda medicine. Though we have the structured data sets and the charak, we have the everything is documented, but like for example in the which we have to connect with the modern medicine, so that we need to think about how we can use the Ayurveda knowledge, or how we can connect the Ayurveda knowledge with the modern medicine for the accelerating drug discovery. So this gut microbiome we can consider as a mediator for the identification of the various processes in the disease causing and the maintenance of the health. When we talked about the AI/ML, that is artificial intelligence, machine learning, basically these things are work with the data. So we have a data, but as in the earlier slide, I mentioned that very limited data is there. So once we have a large number of data to prove this concept, we have a data to prove the complex interaction between the herb, microbe, and the human system. This will help us to accelerate the pattern or accelerate the drug discovery by predicting the how the individual is responding to the special any kind of therapy, how this AI can be used in optimize the formulation. Okay, that's it. Any question?
Yes, Dr. Akhilesh sir, ma'am,
Uh,
we are going through the questions which are posted
Mhm,
by our faculty members, and a
mhm.
few of them are chosen from the participants as well. So, Dr. Akhilesh will continue with the questions.
yeah, madam. There are many questions in the chat box, madam.
Mhm.
So the question is from Sheetal. How H. pylori enters colon and what are its effect?
Okay, so to answer this question, ideally H. pylori is not entering into the colon. Ideally, in the normal healthy individual, H. pylori is there. But if we have some imbalanced condition, the concentration of this H. pylori will go on increasing. So when we talked about the dysbiosis, not necessarily the what we can say removal of the good microorganism and addition of the bad microorganism. Sometimes the concentration of the microorganism also important. So in H. pylori infection, because of the environmental condition, the concentration of the H. pylori go on increasing, and that will lead to the infection.
Okay, madam. Next question is from the Priya. How
Mhm.
does cesarean section and normal delivery influences the microbiome?
Okay. So when we talked about the C-section and the normal delivery, like for example, normal delivery occurs through the vaginal route. And in the very first initial slide I discussed, so there are different organ-specific microorganisms. When during the delivery, when the child is delivering through the vaginal route, the microbiome from the vaginal route will get accumulated, or what we can say present with the infant. When we have a C-section delivery, the vaginal microbiome, that is kind of a good microorganism which will not get exposed to the baby. Through C-section, it will get exposed to the environmental microorganism. Like for example, that is the main reason the C-section and the vaginal delivery baby have a different microbiome.
So the next is from Dr. Smita.
Mhm.
Are there any research references for the relationship of gut microbiome and hypertension?
Yes, yes, there are research literature available how this hypertension is linked with the gut microbiome.
There is a paper, and which is shared with all the participant by Dr. Supriya. So you can check out that paper. We are also going to put that paper in the LMS.
So the another question is again from Dr. Rasmita. What are the pathological investigations for gut microbiome derangements?
Okay, pathological investigation, mainly we can do some kind of the gut microbiome analysis. So there are certain panels which are present in the investigational when we do the biochemical investigational or the microbiome analysis. So in the various like for example diagnostic center, we can have this kind of diagnostics available.
So Dr. Rohit has asked, kindly correlate dementia with neuromuscular disruption and gut dysbiosis.
So, sir, I have already mentioned about the Alzheimer and the Parkinson's. This Alzheimer dementia is kind of symptom with the Alzheimer also. So we can consider how this gut microbiome can lead to the gut-brain axis or the dysbiosis or the imbalance between the gut-brain axis. So as this is kind of some signaling cascade, which can have the signaling pathway, which can disturb the signaling pathway from the gut through brain some because of the some neurotransmitter disruption, and that can lead to the dementia. So basically the chronic inflammation, oxidative stress is one of the important causative factor along with the aging.
Jyotsna Nair, she has asked, what is blood-brain barrier disruption?
Okay. Blood brain barrier disruption is that means like for example these microorganisms are actually secreting some kind of hormones and some kind of the metabolites. So these metabolites are uh, not able to pass through this blood brain barrier, and that disruptions is basically there are filters kind of thing when we talked about the metabolites. So when we have this blood brain barrier disruptions, this imbalance will cause, and ultimately this blood brain barrier will lead to the disease condition.
Yeah. Madhuri Bhide, she has asked, do commensal microbes resides on skin and other sites like wound other than gut?
Sorry, sir, will you please repeat?
Do commensal microbes resides on skin and other sites like wound other than gut?
Yes.
Okay. So Dr. Rohit Johari, he want to know about the differentiation between probiotics and prebiotics.
Okay. So basically, when we talked about probiotic, it is kind of a consortia of the organism. We give the microorganism as a supplement or as an intervention. That is a probiotic. Buttermilk is a good example of the probiotic. In the buttermilk, we have a lactobacillus. So that is an example of the probiotic. Prebiotic is kind of a substrate, some dietary material which can provide as a substrate for the growth of the beneficial microorganism. I hope it is clear, right?
Yes, madam.
Okay.
Dr. Smita, she want to know, are there any short-term courses on gut microbiome which the Ayurveda graduates can do?
I think if you can just refer the recent publication, then you'll get to know how you can explore the Ayurveda concept in the microbiome research. So I don't think there is a need to do any kind of the course. You have to just understand the connection between these two, and you have to just think about as a Ayurveda perspective how you can use this kind of the technology in your clinical practice or in your research.
Yes, madam. Madhuri Bhide, she has asked, why the food fiber promote gut microbes?
So, as I mentioned, these fibers are nothing but the prebiotics. So, based on the constituent of the prebiotics, the growth of the beneficial microorganism is determined. Like, for example, if you have a healthy food, that will lead to the good microorganism growth. If you have a junk food, that will lead to the bad microorganism. So, that's why these food fibers considered as a substrate for the microorganism. When we talked about the herbal fibers, which can have a positive impact, which can lead to the good microorganism growth.
Thanks, ma'am. Dr. Smita, she has asked, how can saliva testing be used for gut microbes?
So
Saliva testing for gut microbes.
saliva testing is not directly used for the gut microbiome, but saliva can be considered as a one of the sample for the analysis of the microbiome. So through saliva, we can have the oral microorganism. Like for example, this overall digestive tract starts with the mouth and it ends with the rectum. So when we have some kind of the dysbiosis in the salivary sample, we can correlate this with the gut microbiome. So not directly can be used for the gut microbiome analysis, but yes, indirectly. There are certain evidences the gut microbiome is disturbed, which we can analyze or which we can predict through the salivary microbiome analysis.
Tripti, she has asked how dysbiosis affects the pathology of diabetes. What are the things which can be used as therapeutics? As we have studied that most of the dairy products are contraindicated in diabetes mellitus.
Yes. So here are 2 points are there. When we talked about the dysbiosis in diabetes mellitus, that diabetes or any metabolic condition is linked with the inflammation and the oxidative stress. And I have already explained the pathway how the oxidative stress and inflammation is leading to the cause of any diseases. So diabetes is one of them. Then we talked about the dairy product. So here again, the what we can say, what substrate we are providing to the microorganism, that matters a lot. When you have a buttermilk, that is also considered as a dairy product, that is recommended in Ayurveda also and in modern medicine also for the management of diabetes. But when you have a some kind of ghee or some cheese kind of thing, that can provide a bad substance for the growth of microorganism, and these microorganism are not good microorganism.
Dr. Mridul Chitrakar, she wished to know how do probiotic species differ between curd and buttermilk.
I don't think probiotic species will get differ with curd and buttermilk, but during the process from the curd to buttermilk, possibility of having the different what we can say microbiome or the microorganism species are there, or the concentration may get vary, and the procedure preparation method can also have an impact on the microbiome community.
Tanushri, she has asked function and factors regulating the composition of human gut microbiome.
I think I explained this in one of my slide. Like for example, starting with the birth mode, then diet, exercise, geography. All these factors are very important. Like for example, I mentioned about if you are doing the gut microbiome analysis from the Indian population, and if you are doing the gut microbiome analysis from the US population for a similar disease, it is drastically varied because the environmental factor and the geographical conditions are like that. They have a specific kind of the microbiome.
Aishwarya Ranade, she wished to know where can we conduct microbiome test and what what cost per test, the cost
So
of per test.
okay, so as a patient, if you want to do the microbiome analysis, so there are majority diagnostic center generally usually they offer this microbiome analysis, and approximately it costs 10 to 12,000 per sample. As a research purpose, we can have a collaboration with the various national organization, where in the collaboration we can do this kind of microbiome analysis.
Dr. Praveen MB, he wished to know at what age the gut microbiome establishes and how we should think about the same in infant while treating various diseases. Whether those interventions are having a long-term effect on gut microbiome.
Okay, so this gut microbiome is established from the time of your birth, and during birth, throughout your journey, it will get changed based on your environmental factor, based on your habit, based on your dietary composition. So during childhood, if you have a good healthy lifestyle, the possibility of that having your immune system is more strong as compared to the other part. And what was the next question? Whether those interventions are having a long term effect on gut
Yeah.
microbiome. So
Yes.
this we don't consider these as an intervention. This is kind of a lifestyle. This is kind of a routine practice we do. Generally, we do the intervention when we are treating some kind of a condition. So if you maintain your healthy lifestyle throughout, ultimately your dysbiosis will not occur, and you will have the healthy and good gut.
Okay, madam. Aditya Hatwar, he wished to know, is there any relation between antimicrobial resistance and dysbiosis?
Yes, of course, there is a relation between the antimicrobial resistance and dysbiosis. As I have mentioned, the intervention or the drug type or use of the antibiotics also play a very important role in the dysbiosis. Like, for example, if person is continuously taking the antibiotic dose every alternate 2 weeks, if person is taking antibiotic, that microbiome will got resistance towards that drug, and ultimately this pathogenic organism for which we are taking the antibiotic will just go on increasing because we have already resistance was developed, right? So there is a obvious link between the antimicrobial resistance and the dysbiosis, and we have to think whatever medication we are taking, we have to think how long we are taking this medication.
Okay, madam. Diksha, she wished to know, are there any specific symptoms of gut dysbiosis?
So there are not as such specific symptoms, but you can understand that your gut is not proper. And I think in Ayurveda there are various methods, like for example, bloating, uncomfortable, not feeling good. That is also one of the symptom which we can consider as a indicator for the gut dysbiosis. So mood is also one of the important factor. Which, if your mood is not consistently good, that means there is something bad or something wrong in your gut. So you can correlate this with your gut dysbiosis, some sleep pattern, some dietary habit, what we can say, craving. So these kind of symptoms generally or usually we correlate with the disease, for example, diabetes or any other thing. But yes, we can consider these as a symptom for the gut dysbiosis.
Madam. Aishwarya Ranade, she has asked, can we measure oxidative stress in any system or an organ? Oxidative stress on in any system or organ, can we measure?
Yes, we can check. Like for example, I have given few samples in the biopsy. We can check with the fluid. We can check the oxidative stress level with the biochemical method. But ultimately, few of the biochemical marker, like for example CRP and this, we can just correlate this with the inflammation and the oxidative stress.
Dr. Sarita, she wished to know how to preserve the sample for microbiome analysis. Preservation of the sample.
Okay. So usually this microbiome stool sample or some biopsy samples are stored for the -80 degrees centigrade, but not necessarily more than 1 year. You can just preserve six months to 1 year these samples for the actual representation of the microorganism. If you can preserve it for long time, possibility of having the what we can say misinterpretation is more.
Professor Dr. Aparna, she has put question. What is the connection in between gut microbiome and skin diseases?
So here the gut microbiome and skin diseases, again it will get linked with the your immune system. If your immune system is low, ultimately the possibility of having infection is more, right? So if you have any recurrent or the consistent infection, skin infection, that means that your immune system is not good. You have to work for your immune system, and this immune system is linked with your gut microbiome.
Yes, thank you, ma'am. I think the questions are flowing. There are many questions coming in, but they appear to be of general category that can be gone through the text. So we will be providing few text after this in the Google Classroom, and you can join the Google Classroom, and there you can get answers of most of the questions. though not ready-made. You have to go through the text and find it out. Supriya ma'am, are you here? Would you like to comment on
Yes,
yes,
I'm
how to
here.
think about this? So this is an excellent session by madam, and madam has given many insights, and she has covered. Actually, this is the benefit of madam, that she has worked with you and was contributed in many of the Ayurvedic thinkings and papers. So definitely, we'll be happy if you could share the links of those papers, and we can put them in the LMS so that it will reach to everyone and will remain with them. Uh, Ma'am, we would also like to know anything more you want to contribute, that how it started, the thinking process, and there are many things which may be there, like having the impact of the ahar and vihar and many other factors, and like very of our favorite things like vishas. Then coming to viruddhahar and many other concept. Can we think of that? Can you just have your opinion? Supriya ma'am, are you here?
Yes, I'm here. And before I answer your questions, I just want to congratulate Sarika because she has really made me proud by presenting such a nice talk. So thank
Thank
you,
you.
Sarika. It was really very nice. Uh, Second thing which I would like to highlight few things. Sarika has generated a lot of curiosity among the all the viewers, all the attendees, whatever we can say, and it is going to be a good area for research in future, I guess. We have to understand that Ayurveda has always considered gut as Mahasrotas. And gut microbiome is a link to explore this Mahasrotas concept of Ayurveda. Like gut is now known to have different axes. Sarika has already shown gut brain axis, gut skin axis, gut lung axis. So through gut, how we can address these diseases? For example, in Tamakshwas, we have said, like, it is said that Tamaketu Virechan. So how Virechan works, how it is connected with Tamakshwas. So all these things which are gut rooted, we can explore through this gut microbiome concept. This is something which I wanted to say. The third important thing is for everybody's understanding, I want to tell that DBT has published Indian Human Microbiome. DBT has started Indian Human Microbiome Initiative, and all this information has been published and is publicly available. So anyone who is interested can go through this information. I also want to tell that DBT, that is Department of Biotechnology, has recently launched a call specifically for artificial intelligence and gut microbiome through Ayurvedic lens. So this was a very significant step that DBT kind of institute is also addressing, acknowledging Ayurveda's inputs or Ayurveda's insights for gut microbiome modulation. And there was a call, the first call was released in May or June, I guess. And such calls will keep coming. So thank you, Sarika. You have highlighted the AI part of it also. And lastly, like thing which Sarika has mentioned, that how genetics is important in gut microbiome. So there are people who are working how the microbiome runs in families, whether the microbiome is also transferred from family generation to generation, like skin microbiome or gut microbiome also, how it is transferred from families in generation, transgenerational transfer of microbiome, that is also being researched. So these are few emerging concepts and very interesting concepts which we can study from Ayurveda perspective. And now coming to Mohan Joshi sir's question, like yes, there are so many concepts in Ayurveda which we can study using this kind of using this framework, gut microbiome as a framework, and we can explore many things using this framework. This is what I can just add to the things which Sarika has said. And there are so many questions coming up. So it was really very interesting session. Just I want to clarify one concept, which was a last question, that there is no normal range of gut microbiome. It is not a thing, like it is not a blood level something substance which you can count and there are levels. It is like what is a proportion of good microbiome and bad good microbes and bad microbes in your gut. And in specific diseases which Sarika has highlighted, metabolic diseases, percentage of one specific species, generally a bad, keeps going increased, and then we have to target that and reduce that species. So this is the answer. There are no normal ranges for gut microbiome. uh, Sarika, you want to add something to this?
Yes, ma'am, I agree with you that I have already mentioned that concentration matters. Microorganism concentration matters a lot for the causative as a causative factor.
Yes, so this is all from my side, Mohan sir. And last thing which I probably would like to say that sometimes we need or we want ready-made answers for everything, but it is not possible because neither me nor Sarika, we have worked extensively in this area. There are few papers, but we all collectively can work and find out answers to certain questions, how it can happen and how it can work. So I think this is the first step, and we must congratulate All India Institute of Ayurveda Goa for taking this step. And so many interesting discussions have initiated. We will take them forward. Thank you, sir.
Yes, thank you so much, ma'am. So before we officially close, I would like to thank madam, and I would like to ask the participant. So this is a question directly to the participants. In the recent many of our lecture sessions, we have seen that there are hardly 2 or 3 questions coming up, and there is no much interaction in between. But this topic and the presentation itself was so interesting, and the topic is something which attracts everyone because it is connected to our core concept of giving importance to the agni, giving importance to the digestion, and relating it with every part of our disease and metabolic system related to disease development, and again to the treatment. So questions, certainly there will be questions that how anything ask. Now put this as a center point, and if we start on asking the questions, how Samsarjan Kram works, how Vamana works, how Virechana works, how Basti works, how drugs works. So there will be n number of questions coming up, and probably uh, as Supriya ma'am said, and this is something a initial stage, and we have to find out as many as answers, and probably it will uh, just unfold the principles in a better way in front of us, so bringing the validation for our own concepts. So that's how we should think of it. uh, I think we can close. Sir, would you like to summarize and we'll close?
Dr. Sarika ma'am, I express our heartiest gratitude to you ma'am for sparing your valuable time and delivering such an enlightening lecture. Madam has given detailed explanation from the basics. What is microbiome? A hidden organ, factors influencing composition and function, gut dysbiosis, therapeutic potential, future direction, techniques for studying the microbiomes, Ayurvedic linkage, publications. She has also explained Ayurvedic herbs and diet as microbiome modulators and future collaboration. We are truly grateful for your presence and contribution today, ma'am. Again, I am thanking Dr. Supriya Bhalerao ma'am for providing us with such an excellent speakers and constantly being in touch with us. And thank you, ma'am. Thank you to the organizers, our Dean ma'am, Professor Dr. Sujata Gadam madam, our Associated Dean, Dr. Mohan Joshi sir. Thank you for arranging such a wonderful webinar. Thank you all, the participants, for your patient listening. And we will meet again tomorrow at 7 p.m. sharp. Thank you.
Thank you, sir.